All right, we have finished discussing about linkage, and in linkage we have already talked about how when the genes are close to each other, it is very difficult for them to be separated. They are transferred from one generation to another, almost always together. In this particular topic, that is crossing over, we are going to discuss about genes that are located at a distance. As discussed earlier in Morgan's experiment, when the genes are located at a distance, there is every chance for a recombination. And recombination basically means the mixing up of the genes. So in this particular section, crossing over, we're going to talk about how this mixing of genes takes place. How does recombination take place? The process by which there is genetic exchange between homologous chromosome is called crossing over. The process by which there is genetic exchange, that means if these are two chromosomes, the process by which the gene A and gene A, they exchange with each other. When we're talking about crossing over, one thing we got to remember in mind is that we're talking about homologous chromosomes. And I'm sure you know what is homologous chromosomes. Homologous chromosomes are almost of the same size, structure, shape and function, and they carry the same genes. One half of it comes from the father and the other half comes from the mother. That is basically what is homologous chromosomes. So when we are discussing about genetic exchange, we're talking about genetic exchange between homologous chromosomes. We are not talking about non-homologous chromosomes. So it is very important, we got to remember that when we're talking about genetic exchange, it is the homologous chromosomes that is taken into consideration. And when does this type of crossing over take place? It basically happens, there could be genetic exchange in the mitotic cell division as well as meiotic cell division. When the genetic exchange takes place in mitotic cell division, that change that is there, it could be in the phenotypic change, it could be represented by a phenotypic change as well, but that change is limited to that particular person, only to that particular generation. It is not transferred from one generation to another. So basically when we're talking about mitotic genetic exchange or crossing over, it is limited to that person with whom that genetic change has come into place. But when we are talking about meiotic genetic exchange, or we can say meiotic crossing over, then it is transferred from one generation to another. Now here we are going to discuss about meiotic crossing over that takes place during meiosis. As we already know, meiosis has two phases, meiosis 1 and meiosis 2. We already know the fact that meiosis 1 is a reduction division and meiosis 2 is more or less a mitotic cell division, mitosis. So we are limiting our discussion to meiosis 1 and especially to a particular meiosis 1, the prophase of meiosis 1. Prophase 1, because it belongs to meiosis 1. We are limiting our discussion to prophase 1, during which in the Pachytene stage, this prophase 1 has been divided into a number of stages. Liptotene, zygotene, Pachytene, diplotene and dikinesis. Here we are talking about prophase 1 and particularly during Pachytene substage. Pachytene substage. We are talking about the Pachytene substage. This is basically when the chromosomes are in the form of dyad. Dyad means they have 4 arms. Dyad means 4 arms. So basically we got to take into consideration that chromosomes, when they are in pairs, they are as monads. That means these two chromosomes, these are in monads. That means they have only one arm. When we're talking about dyads, we're talking about chromosomes with four arms. This is a homologous pair. So the homologous pair of this will also have four arms like this. Okay, this is a dyad stage. The process takes place in the Pachytene stage when the chromosomes are in the dyad stage. That means it is formed after the replication of chromosomes. That means during the synthetic phase, when the replication of chromosome takes place, then this extra arm comes up, which are exactly same to each other. These arms are called now the sister chromatids. These two arms of one chromosome are called the sister chromatids. Same here also, these are called the sister chromatids. And likewise, these two arms of two chromosomes are called non-sister chromatid. So basically, crossing over takes place in the non-sister chromatids of the homologous pair. The non-sister chromatids of the homologous pair. Here they are in dyad stage. Now when crossing over takes place, let us understand it with the help of a diagram. And when we're talking about crossing over, we're basically talking about the nuclear activities that takes place inside the nucleus with the chromosomes. Okay, so let's take for example a cell with a nucleus and it has two chromosomes, two chromosomal pair. Alright, they are in which stage right now? They are in monads. Alright, now during the interphase, during the synthetic phase, the chromosome doubles, the chromosome number doubles. The chromosome, the DNA doubles, not the chromosome, the DNA doubles. So it enters into another stage when they look like this. They are in dyad stage. Alright? So in prophase 1, during the Pachytene stage, what happens is this homologous pair and their non-sister chromatids overlap with each other. The next stage that happens here is like this. So they overlap like this. They overlap like this. This overlapping is called synapsis. This overlapping is called synapsis. It is formed by a synaptomal complex. Synaptomal complex. During the Pachytene stage, they overlap like this. What overlaps is the non-sister chromatids. The non-sister chromatids overlap with each other. Now to understand it in a better way, this overlapping will, draw a diagram of only of the chromosomes. I will draw only one pair. This is the dyad stage. Now, when the overlapping occurs, it looks like this. Since they are homologous chromosomes, they are of the same size, shape, and color. But to make it more prominent, we'll just make this one of the pair as white, so that we can understand it better. We'll make this white. Alright, so this is the homologous chromosomes. They are in what stage? Dyad stage. Okay, now when synaptomal complex occurs, or we can say synapsis occurs, then they look like this. They are in dyad stage. the non-sister chromatids overlap with each other. They overlap here. This is called chiasmata. They overlap like this. This particular, when they overlap, the dyads, they overlap, then it is called tetrad. This particular stage is tetrad stage, during which the homologous chromosome, the non-sister chromatids of the homologous chromosome, overlap with each other by formation of chiasma. After this stage, what happens is, when the synaptomal complex or the chiasma moves like a zipper and the two chromosomes again separate, then something of this phenomena occurs, in which a small segment of this one comes over here, and the small segment of this one comes here. So there is a slight exchange of genetic makeup. This is what is called the crossing over. And this crossing over requires several conditions. The first condition is the two homologous chromosomes must pair and form synapses. That means they have to pair and form synapses. This homologous chromosomes. And the homologous chromosome must be present in a tetrad condition. This is the tetrad. These are dyads. When they cross over, when they overlap, then it is called the tetrad. And for crossing over to take place, the genes has to be located at a sufficient distance. They cannot be close together. If the genes are close together, then chance of crossing over is much less. But when they are at a distance, then the chance of crossing over is increased. The maximum percentage, or we can say the maximum value of crossing over is 50%. That means there could be a 50% chances of crossing over. So these are the conditions that we need to remember. There are three conditions. Two homologous chromosomes must pair to form synapses. Homologous chromosome must be in a tetrad conditions, and genes must be located at a sufficient distance for crossing over to take place. So the maximum percentage of crossing over is 50%. Now we move on to the significance of crossing over. What are the significance? The first significance is the crossing over due to small exchange of genetic makeup brings about variation. When we talk about variation, let me just explain you a bit. We have half of the chromosome coming from our mother and half of the chromosome coming from our father. Right? We should look exactly like our father and mother, but we don't. There are certain characteristics in us which are neither of our parents, which means that we are slightly different from our parents. That is what is called variation. And this recombination of genes brings about, slight exchange of genes brings about variation. And remember that this variation is a very important raw material for evolution to take place. From the values of crossing over, what percentage of crossing over has taken place, we can determine the distance between the two genes. We can approximately determine the distance between two genes. That is the gap that exists between the two genes, on the basis of which genetic maps can be created. In fact, we'll be talking about this in the next section when we talk about Human Genome Project. Another important significance is there exists a relation between the percentage of crossing over and their distance. The percentage of crossing over depends upon the distances between the genes. That means one genome project. Another important significance is there exists a relation between the percentage of crossing over and their distance. The percentage of crossing over depends upon the distances between the genes. That means 1% crossing over is equal to 1 map unit. 1% crossing over is equal to 1 map unit, which is represented as mu, which is also equal to, or we can say this is also called, or it is also called centimorgan. Centimorgan. This unit is also called centimorgan or map unit. And the percentage of tetrad involved in an exchange between the genes is twice the percentage of recombinant gametes produced. So these are the things which we need to remember when we talk about crossing over. So much for today. We'll carry on with hereditary diseases in the next segment.